That's the same. Is it the polar phosphate head group? Oh yeah, that can be different. Can the fatty acid tails be different? Yeah, they can be different. So they can have different numbers they can be different lengths, they can be saturated or unsaturated. And what does the difference between being saturated and Rita asking something? Yeah.
So those three components are in all, but only the glycerol backbone is same.
That's right. The glycerol backbone is consistent. And of course, the general idea of having a polar phosphate head group and two fatty acid tails is the same, but the specific structure of those can vary. Right? And so when you have saturated fatty acids, two saturated fatty acid tails versus an unsaturated fatty acid tail, what does that mean for the qualities of that membrane? Yeah, so if you have unsaturated fatty acid, what does that do? Maybe someone want to raise their hand? So double bond is unsaturated, and structurally, what does that do? That's when it's saturated, right? If it's unsaturated, that kink causes space between the necks. More fluidity. You got it. So unsaturated fatty acids are associated with membranes being more fluid. You have more, they can wiggle around, or there's a little bit more space. Whereas saturated fatty acid tails means they can jam in like sardines, right? And they're less fluid. Okay, and then the big take home, right, was that there's variation in the lipids, and one of the variations that's going to become really important as we start to talk more about vesicle formation and fusion with other membranes is the fact that you see differences in the two leaflets. And by leaflets, right, it's a lipid bilayer, there's two layers. Each one is referred to as a leaflet. And one leaflet is going to be referred to as the non-cytosolic side, right? And one is going to be called the cytosolic side. So in a lipid bilayer, there's always going to be one side facing cytosol, and then if it's the plasma membrane, one side facing the extracellular space. If it's a vesicle or an organelle, it's going to be facing, the non-cytosolic side is facing the lumen of that vesicle or organelle. Right? So whatever's facing the cytosol is called the cytosolic side. So we'll come back to that, and now we're going to kind of talk a little bit about some general features of membrane proteins. So first and foremost, the proteins that reside on the membrane, if you're talking about the plasma membrane, they typically are regarded as lending that cell its identity. Right? So cells you can recognize by what proteins are on the surface of those cells. So none of you have taken immunology yet because this is a prereq for it. However, there are a couple cell types that are prominent players in our immune system called T cells and B cells that you might have heard of before. The way that you can tell the difference between those cells is the surface proteins on the cells. There's this cool technique that you can use in laboratories where that allow you to isolate. Like for example, this is used in stem cell biology all over all the time. It's called flow cytometry. And flow cytometry uses this machine that has lasers that can identify basically specific fluorescent profiles, and it totally hinges upon what are on the surface of the cells. That can give it, after you add fancy reagents, a different fluorescent profile. So have you guys ever heard of CD markers? Sorry, I'm getting very technique-y here, but this is cool. CD markers, it stands for cluster of differentiation, and all cells have different CD markers. And so when scientists are trying to isolate cells from a population, the way they do it is get to know what are the specific CD markers on that cell, and then they use a flow cytometer to separate them based on a fluorescent profile. That was a long way of explaining this kind of fragment here. The variation in the proteins on a membrane are thought to give cells kind of their identity. It's a way that you can separate one cell from another. They have different proteins. And all these proteins are associated with specific functions. And on this slide, the other idea that's trying to be relayed to you is that these proteins can be associated with membranes a bunch of different ways. So you can have transmembrane proteins, right, that the whole protein itself spans the membrane. And so they come in different flavors. You can have a single pass transmembrane protein, so it just passes through the membrane one time. You can have multi-pass, which winds back and forth across the membrane. So these two have a difference in the number of what? Yeah, you got it. The difference in the number of start-stop transfer sequences. That's right. That's what tells the protein translocator to embed that stretch into the membrane. And then there are also proteins that are barrels that serve this barrel or channel like function. There are also proteins called single sheath proteins. These, right, single sheath, because they only pass through one of the, they're only integrated in with one of those bilayer leaflets or sheaths. Some proteins aren't, there's no like amino acid stretches that are actually associated with or embedded into the membrane. They're attached via other molecules. So this would be an example of a lipid-linked protein, right? So here's, the green is the part of the protein that's composed of amino acids. Pink would be covalently attached lipid that stick itself right into that membrane. Sugar anchors, so in some cases there's an oligosaccharide that is what helps anchor the protein down. And then there are peripheral membrane proteins that are associated with the membrane by being stuck to transmembrane proteins. So they are next to that membrane and they help give that membrane its identity and serve some important function, but the protein itself, the way that it's stuck to the membrane is through an association with a protein that's stuck to the membrane. So basically, if you can think it, a protein is associated with the membrane that way. All these different variations in how they can associate with the membrane. Okay. Yeah?
Sorry, can I comment outside? Can you explain why 4 is not considered transmembrane again?
Yeah, so
Right, trans goes different.
You got it. I'm sure.
Class.
How dare you apologize? It's my choice. How dare it's their time too. I'm sure there was someone else thinking the same thing though. So, yeah, we got another question.
For can it only is it only like extending into the cytosol, or can it also extend on the?
Oh yeah, you can have single sheath going either direction. Yeah. All these, yeah, go kind of every which way. Okay. So some of the typical functions that you see in transmembrane proteins, they're transmembrane proteins that function solely in transport. They transport things from one side of the membrane to the other, right? Not everything can get, in fact, most things can't get across on their own. There are membrane proteins that function as enzymes. There are proteins in the membrane that function in signal transduction, so they function as receptors. Yes, they can have both functions at once. Things get wild. They can function in cell-cell recognition, so they help recognize another cell. That becomes really important too in immunology. One of my other favorite subjects. Intercellular joining, so right when we talk about cells adhering to one another, there are proteins that are relied upon to link cells together. We'll go into that in greater detail in our cellular adhesion lecture. And then adhesion to the ECM, there are receptors also that are responsible for attaching to proteins in the extracellular matrix. So these are 6 of the most common functions that you see in membrane proteins. And one phenomenon that's interesting is there is a lot of variability in terms of the ratio of proteins to lipids in any given membrane. So a myelin sheath, right, the myelin sheath, its primary role is just to function as like an insulator along that axon, right? So you don't really need a lot of membrane proteins. You just want it to function as something that wraps around and around and around. And in fact, you see a relatively low amount of protein in the membrane that composes that myelin sheath. Way more heavy on the lipids. Outer mitochondrial membrane is about 50-50, which is about the ratio you see in most plasma membranes, about 50-50. Inner mitochondrial membrane, 80-20, way heavier on the protein. So this is interesting. So if you think back to the endosymbiosis, that theory, right? Where does it say that mitochondria come from? Or where do mitochondria come from? From a bacteria that was engulfed, right, by a primitive and ancestral eukaryotic cell, right? And so that inner membrane, when that cell engulfed it, the outer membrane would have come from the eukaryotic cell that engulfed it, right? It would have surrounded it with membrane, so it would have been plasma membrane. The inner membrane would have come from bacterial membrane, right? Guess what ratio of protein to lipid you see in most bacterial membranes? Yeah, 80-20. Pretty cool. I was so cool. Blowing your mind already. 14 minutes in. No. You guys need seatbelts. Okay. So let's talk about sugars, right? We talked about oligosaccharides being really important as well. And so oligosaccharides we know are certainly important for the way proteins fold. And it turns out that oligosaccharides that are on proteins, particularly in the plasma membrane, are what are part of what confer like cellular identity to a particular cell. Like it's part of the way our body recognizes those cells. And if that's still confusing, I think an example that helps make it make sense is when we talk about the blood grouping, right? So we all know that if you get a blood transfusion, you want to make sure that the blood type is matched, or you get O blood, right? So the reason for that is because these represent antigens that are recognized by antibodies. So your body and your immune system, one of the most important things it does is differentiate between you and things that are foreign, that are not you, right? So I have blood type A plus, actually. Hi, Chief. I thought that was super cool when I was, anyways. Obviously, I still feel cool as I'm carrying it. But type A blood. So the antigen on my blood is the antigen A. So my body recognizes A as self. So people who have blood type B, that antigen looks different. So if I was to get a transfusion of type B blood, my immune system would recognize that as not me and would mount an attack, right? The difference in those antigens, it's a difference in the structure of the sugar that's on the surface of these proteins in the red blood cell. So the difference comes from a glycosylating enzyme. It's called the ABO glycosyl transferase. And there are different versions of it, right? That means that there's variety in the way that those sugar groups look. So people who have blood type A, the sugar groups that are presented on the surface of their red blood cell have one structure. People who have blood type B has another structure. Blood type AB, guess what? Both structures, right? So if you have both structures, cool. If you're type AB, that's really the best blood type because you can get any blood transfusion that works with you because your body recognizes A and B as self. Blood type O, guess what they have? Neither antigen, right? So if you have blood type O, you can only get blood type O transfused to you, but you can give your blood to anyone.
Oh, thanks.
Does that make sense? Okay. All comes down to a difference in the structure of that oligosaccharide. So the other important aspect of sugars that I want you to appreciate is yeah. Does that follow? I do not know about Bombay blood type. I feel like this is when my son first realized I didn't know everything. I don't know everything. One time Emmett said, "Mommy, do you know all the things?" And I wanted so badly to say yes. And yeah, just be superior when my kid was 4. But then I was like, no one knows everything, buddy. But I know, so there's the rhesus factor. That's a little bit different. That's an actual protein. So I don't know if Bombay factor is similar. I'm obviously going to look it up in my quest to know everything. So that someday I'll be like, I know everything. It's not possible. If you understand science, you know there's no way you know everything. People who act like that waste the time to continue a conversation with them. Yes. All right. Okay, so this slide is going to explain why sugars are always going to be found on the non-cytosolic side of a protein. So, where did I mention, and there are exceptions to this, but I can talk about those after I teach you the rule. So, where is oligosaccharyl transferase? You guys remember that enzyme or what it does? If you want to raise your hand, that sounds like,
Is it in the membrane?
It's in which organelle?
Not sure.
Okay. I appreciate you, Ron. Okay, so let's break it down. Oligosaccharyl transferase. It transfers what? Oligosaccharides. And it does it to asparagine residues. Sugars. Where does that process happen? That in-link glycosylation. You guys remember? It's in the in-link, and it happens in the ER. I'll just tell you. I don't have all these people. Just kidding. Make sure you know before the test. Make sure you know before the test. So it happens in the lumen of the ER, right? So let's pretend in this image that we're looking at the lumen of the ER. And here we have a transmembrane protein that's been co-translationally translocated, of course, into the membrane. The oligosaccharyl transferase is going to add the sugar to the non-cytosolic side of that protein, okay? So the side that's facing the lumen. So that's the first thing you have to accept or understand, right? That the oligosaccharyl transferase is going to add that in-link sugar in the lumen of the ER, okay? And remember that lumen's non-cytosolic. Cytosol's out here. Non-cytosolic, lumen is here. So here's where the sugar is going to be, right? So say this protein is ultimately going to the plasma membrane, or it's going to go to the Golgi next, and then the plasma membrane. So the way that vesicles bud, and we're going to come back to the same image again pretty soon, is they pouch outward, right? They get pulled into this bud. So we have red and blue here, which represent, what do you think red and blue represent? So it actually representing the 2 leaflets of the lipid bilayer. And the reason they colored them differently goes back to that very first slide I showed you there, that the lipids in the inner layer are different, have a different composition than the lipids in the outer layer. Okay? So when this buds out, remember the cytosolic side, in this case, of the lipids is red. It remains cytosolic, right? The way this membrane pouches outward, the cytosolic leaflet remains cytosolic. Non-cytosolic remains in the non-cytosolic luminal facing side of the lipid bilayer, right? Okay, so that protein that has the sugar here, it's still facing the lumen, right? It pulls the protein down, and so the protein is still facing the inner core. You guys with me so far? Okay. And here's the only part that, well, not the only part. Here's the part I think people usually get a little bit lost on. As this vesicle travels, right, it just maintains that conformation. But when it fuses with its target compartment, the way that fusion proceeds is the non-cytosolic leaflet, it wants to fuse with the layer that is similar to it, right? So the red outer cytosolic leaflet in this case is going to fuse with cytosolic leaflet. And then the blue non-cytosolic inner leaflet will fuse with the blue non-cytosolic inner leaflet. So what happens then is that fusion occurs, it causes outer to fuse and inner fuses, and it causes that vesicle to basically fuse like this. Okay, and when that happens, that means that it dumps its contents into the lumen of the target compartment, or if it's fusing with the plasma membrane, that would represent the ECM. Yeah. So get back to this protein that was glycosylated on the non-cytosolic base. That non-cytosolic orientation is preserved when it's fused. So that sugar that was added here stays facing non-cytosolic side throughout vesicle transport and diffusion. And that is something that you need to really learn to make sure you have it. These are yeah. Yeah. So when this fusion occurs right here, I'm gonna be dramatic. Just woke up and just sleepy. Go sleep again. Okay. So basically, what I'm saying is, so we're gonna do red and purple instead of red and blue.
Just mark today. I mean, sure.
Okay, now we want fusion. Okay. So the way this fuses is that membrane fuses so that you still have the same orientation. What you do not see you never see that. That does not happen. So the orientation of the way that the lipids fuse stays Any sense? Right. Right. So to get that, you have to make sure you understand that the lipids are different. They're not just different colors, right? This is just a proxy for the lipids, the fact that they're different. So you have a protein in here that has a sugar group on it. When the sugar group is closest to the non-cytosolic leaflet, right? So when this fuses, it maintains that conformation. Right? Non-cytosolic stays non-cytosolic. It's totally dependent on the mechanics of that lipid fusion. Okay. So I have two head nods and fewer furrowed brows. Yeah.
So the sugar is found on the transmembrane.
Yeah. In this case, the example was of I was kind of talking about transmembrane proteins, and this is also why you only see in-linked glycosylation on proteins that move through the ER. Not all proteins move through the ER.
Dr. Modi?
Yeah.
So is the ER usually shrinking?
What?
Like,
No, the ER is constantly making new lipids. So lipid synthesis happens in the ER. Yeah, good question. And you kind of constantly have vesicles forming and fusing all over the place. So, yeah.
So for the donor, how would it get rid of, for the recipient, how would it get rid of the extra lipid that you accumulated?
Oh, it's just that every single lipid-containing organelle is constantly undergoing, like there are vesicles fusing with it and vesicles leaving from it.
Okay.
Yeah, so kind of any membrane might have some like net growth or net loss at any one time, but it's pretty much maintained. And I'm sure there's homeostatic mechanisms that would induce or limit lipid synthesis as needed.
Okay.
Yeah.
So does that mean that on the inner part of the membrane, OLC will never be affected on that,
Oh, right. Yeah, and that was the thing that that was a question that I actually want to investigate because I know of instances where there are lipid-linked membranes on the non-cytosolic side of the plasma membrane, and I think it's because there are dedicated proteins that hook them. Because there are these flipases, flopases, scramblases that help move things around. It's just you don't see that happen with big old transmembrane proteins. Or so far, right? And I'm sure there are exceptions.
And this only applies to the ER membrane, right?
That's right. You don't see in-linked glycosylation anywhere else in the cell. So in-linked glycosylation leads to the addition of these big old oligosaccharides, specifically to asparagine residues. There's another type of glycosylation that occurs in the Golgi called O-linked glycosylation, and it seems like to some limited extent, there is some very minimal O-linked glycosylation in the cytosol, but it's these really tiny sugar, more like sugar monomers, so it looks structurally really different. Yeah. Sorry, that gave me a thought that I'm not going to share because it's way it's that topic. But I think there was another question on there somewhere. Okay. All right, the same slide we're coming back to a few times, so if you're still confused on this, you can shout out your question. I mean, anytime. Really. I'm always fine if you guys are like, hey, I have a question, it's related to something 5 slides back. I'm happy to go back. If you haven't been able to tell yet, I'm more concerned with making sure you know what I do go over than racing through everything. So I'm okay with slowing down if you guys have questions. Less is more. All right, so another component of all biological membranes is really important, is cholesterol. So cholesterol is a sterol with a polar head group. So sterols often have this 4-ring structure, so it's this kind of typical 4-ring structure. This red group is I think denoting the polar, or the hydroxyl group that makes it a sterol. So steroids always have this 4-carbon ring, and then what makes this not just a steroid but a sterol, the OL part, is having the hydroxyl group. More biochemistry than you actually need to know. The part that's most important for you to understand that it also has this hydrocarbon tail, right? That's what this thing that's hanging off of it represents, right? So this hydrocarbon tail, non-polar, hydrophobic, and the thing that's important to know about this 4-ring structure is it's really rigid. So it's not floppy like a fatty acid tail, it's rigid. And so cholesterol can help confer rigidity to membranes. Cholesterol, oh right, it's a rigidity, it makes them more rigid. So the more cholesterol, the more rigid. And so like anything in the cell is always trying to strike a balance. You want some cholesterol, but not too much. And so cells are constantly trying to regulate the level of cholesterol in their membranes. And then the submembrane protein network I mentioned. So here's an image of just the plasma membrane. Submembrane protein meshwork exists on all membranes, and it's regarded as the cytosolic side of the membrane that is just under the membrane and is composed of a bunch of proteins that are more peripherally attached to the membrane. So, different than the membranes that are actually spanning the membrane, submembrane protein meshwork would be the things that are peripherally attached, associated with those membranes. These red tentacle-like things are representing cytoskeletal filaments, specifically actin filaments that form this cool meshwork right underneath the membrane. And so it turns out that the cytosolic submembrane meshwork is really important for kind of linking what's happening with the membrane to the rest of the cell. And one example that we'll talk about in the next lecture is how on vesicles, right, if you have a vesicle, the submembrane protein meshwork is going to be on the cytosolic side of it, right? So on the outside of the vesicle. And it's that meshwork that contains the proteins that link up to the motor proteins that help move the vesicles around. And so I'll come back to this idea of submembrane protein meshwork when I get there. So in this slide, the actual submembrane protein meshwork, what I kind of circled in hash marks here. It's all the proteins and molecules that live just if it's the plasma membrane, just under the membrane on the cytosolic side. If you're talking about organelles or vesicles, it's going to be just on top of the membrane on the cytosolic side. It's always on the cytosolic face of the membrane. And I say that just because I think sometimes it's intuitive when you're talking about the plasma membrane, that this is the cytosolic side. And so we're always thinking about, okay, just inside of the membrane. But you're talking about a vesicle or organelle. It just takes a little bit of mental gymnastics, right, to remember that the outside is the cytosolic side. That's the side that's facing the cytosol. You just have to keep track of that when you're thinking about the next two lectures. So the rest of the slides in that lecture are kind of the review of Bio2, so I don't go over them in class, but make sure you remind yourself of the functions of all those organelles and systems that are there. It is important that you know them. I will throw you a bone and have a very easy question about one of those on the test. That is just recall. Okay. So on to the lectures on intracellular transport. It's really cool, right? So this is talking about how molecules move within a cell. So we've talked about transcription, translation. We have these proteins now. How do they get to where they need to go? And so this image, right? I have this image in the learning module as well, kind of a different version of it.